Please join me in welcoming to the Distinctive Voices podium, Dr. Stacy Kopp.
Thank you so much. It's really a privilege to be here, and thanks to all of you in the audience and the invitation to share my research today. So I'm going to be describing a specific project in my research lab that focuses on the material you can see in the background of the slide here. We are materials chemists, physicists, and engineers, and we study how we can use molecules to make new materials, especially those that make and manipulate light. And recently, we've had a big effort in making new types of light-emitting molecules that could enable deep tissue imaging in the near-infrared spectral region. But to begin the story today, I'm going to begin with light and how light interacts with matter. So for most of us, the primary way that we experience the world is through sight. We are seeing the way that light, in the form of photons, these are the quanta of light, are interacting with the materials that our world is made of. And often, if you're like me, your first experiences with science probably involved seeing things. So for me, seeing light and color was one of the things that sparked some of my early interest in science, and it's really why I'm here today. This picture on the screen you see here is actually from the mountains where I grew up. I grew up in the foothills in New Mexico. And being able to see the way that light would interact with the landscape, the clouds, how rainbows would form, and how plants could transform sunlight into energy, these things inspired me. And we see many beautiful examples of complex light-matter interactions in nature. For example, there are organisms that can really rapidly and eloquently transform the way that they appear by tuning how their bodies interact with light. So this cephalopod is within just 2 seconds transforming from something that looks like a rock to something that is clearly not a rock. And it is doing this by tuning both light scattering and light absorption dynamically. Now, here's another organism that does this that we don't have in Southern California much, but some of you may have had the privilege of catching fireflies. And so you may know that these bugs will glow because they have a biochemical reaction that occurs in their abdomen. What you may not know, and what I didn't know until somewhat recently, is that these bugs are also harnessing nanophotonics. Specifically, their abdomen is coated in nanoscaled patterning, and that's perfectly designed so that it couples to the wavelength of light that the bug is producing and then scatters that light more efficiently. So this bug is harnessing both light generation and light scattering to control and manipulate light. Now, we can also use light ourselves as humans as a tool to understand nature, and there's perhaps no more eloquent and versatile example than fluorescence imaging and fluorescence microscopy. So this is a tool where we take tiny molecules or nanoparticles that can glow, and we label specific parts of a cell or a tissue or even an organism, and then we selectively visualize different parts of that cell or tissue in order to create nanoscale maps of how our bodies and cells are arranged. And this technology has really transformed our understanding of biology. You can watch in real time as cells divide, as viruses invade a cell, or as cargo is carried along by tiny molecular motors inside of the cell. And all of this is by harnessing selective imaging of specific colors of light. Now, you may have not spent hours and hours in a dark room in front of one of these microscopes like I have, but I'm sure that you have all experienced fluorescence and actually have some understanding of how it works, even if you might not know it. So many of you have probably been under a black light and seen how your clothes might glow, especially your white or lighter colored clothes. So what's happening here is that black light is producing UV light, which your eyes primarily can't see. But that UV light gets absorbed by certain molecules or materials, and then those will glow or fluoresce at a different wavelength, and those are wavelengths your eyes can see. So just like in this fluorescence microscope where you can selectively image specific parts of a cell by imaging specific colors, here your eye is seeing the glow from these certain materials or molecules that are in the world around you. Now, what exactly is happening in fluorescence at the electronic level? Here, we can use what's called the Jablonski diagram to understand this. So a molecule or a material by itself will always be in the ground state. This is the lowest energy state, all of the electrons in that lowest energy state. But sometimes, if it absorbs a quantum of light, a photon, one of those electrons could get promoted from that ground state up to an excited state. And now that system wants to return back to the ground state because of thermodynamics, but there's a process that needs to happen. And first, what happens is what's called non-radiative relaxation, or that's a fancy way of saying usually the molecule wiggles. So it's going to vibrate or wiggle, and it will start to lose energy. And then sometimes it may end up in a state where it can then relax down to the ground state by emitting another photon. And because it already lost some energy to that wiggling, that energy of the photon that is produced by fluorescence is going to be at a different color than the light that was absorbed. And this is the natural shift in color that is involved in fluorescence. And this is why it's such a powerful tool for biology, because we can selectively excite at one color and then selectively detect at another color of light. Now, what are these things that are doing the fluorescing? Well, there are many things that are fluorescent. One example, which you might have experienced if you went to get an eye exam, you had your eyes dilated, is fluorescein. So the doctor will put a droplet of some greenish stuff in your eye. That greenish stuff is a green fluorescent dye. Do we have any folks from Chicago here? Maybe not. Okay, so some of you might know that they also would dye the Chicago River green on St. Patrick's Day using this exact fluorescein. So this is what fluorescence is. Now, let's say that we want to use fluorescence to image deep inside of a tissue instead of just a single cell. Well, in this case, we need to think about all the possible light-matter interactions that can occur. So let's say that we want to image that little red star down there, but it's buried in a bunch of other biological tissue. Well, unfortunately, the fluorescence of that little red star is not the only thing that is going to happen. We can also have reflection of our excitation light from the surface of our tissue. That light can also be scattered in other directions away from the red star. It can be absorbed by stuff, and then that will just lead to heat or vibration. So it also won't end up lighting up that red star. And then finally, we can induce fluorescence from other things we're not interested in. So our bodies do have chromophores that will naturally fluoresce, and this is what's called autofluorescence. And all of that gets in the way of seeing that red star that you really want to see. Maybe this is a biomolecule or biomarker of interest. Maybe it's part of a tumor that you've labeled. So how do we overcome this challenge of imaging deep inside of tissue? Well, one way we can do this is by using more of the electromagnetic spectrum. So the light that we see, this visible light, is really only a small sliver of all possible electromagnetic waves. And we know that we are not transparent. If you look at your neighbor, fortunately, they're reflecting a lot of visible light. Otherwise, they would look very bizarre. But if you could see in the near infrared at slightly longer wavelengths, they might look a little creepy because you might be able to see deeper into their bodies. And so it is this spectral window that could give us a deeper window into biological systems. So why is this? Well, there's several different factors that all come together. One factor is that scattering becomes much less efficient at these longer near-infrared wavelengths. So you can see in this graph here the different scattering coefficients of different types of biological tissues, from skin to brain tissue, which is extremely light scattering. And that's one challenge why brain imaging with light is so difficult. Another thing that's of favor here is that our bodies absorb much less in the near infrared. So for example, hemoglobin, both oxygenated and deoxygenated, has absorption peaks in the visible spectrum, but not in the near infrared. So these two things, less light scattering and less light absorption, mean that we can penetrate with light, non-hazardous near-infrared light, much deeper into biological tissues. And then there's a third factor, which is also in our favor, and that's that this autofluorescence, this natural fluorescence of stuff inside of our bodies diminishes much more in the near-infrared window as well. So all three of these things combined would make it seem that fluorescence imaging in the near-infrared window would be a promising strategy to get around this challenge of using fluorescence imaging deep inside the body. So why is it that when you go to the doctor, you have to get an X-ray, an MRI, or an ultrasound instead of using near-infrared light? Well, one of the challenges remains that we don't have good contrast agents that work in this near-infrared spectral window. In other words, we lack small, bright, non-toxic, and biocompatible near-infrared light emitters that we can use to tag and track targets of interest. And our options so far are limited, but we can put them all on this two-axis graph of near-infrared brightness. We obviously want more brightness, and size. So why is size important? Well, imagine if you're trying to label a biomolecule and follow it around, and you label it with something giant. Its behavior is going to change significantly, probably like this toddler attached to this giant dog. So this is why we also need to care about the size of our contrast agent. Now, our options generally fall into four categories. We have organic dyes, like that fluorescent I told you about. These are small. The chemistries to covalently link these to biomolecules has been very well developed, but one challenge is that they're just not very bright near-infrared emitters, and that's for a fundamental photophysical reason. The same challenge affects what are called fluorescent proteins. So these are fluorophores that can be endogenously expressed by an organism without having to add the contrast agent. But again, those have these same fundamental limitations for near-IR brightness at the moment. So our other options are much brighter, but also much larger. And these are typically nanoparticle emitters, things like carbon nanotubes and quantum dots. While these are bright near-infrared emitters, they are larger, and they can also often be quite toxic, containing things like cadmium and lead. So these are also difficult for biological applications. So what can we do to get to that region there, where we have a very bright emitter that is very small? Well, this is the vision that we are trying to enable. We want to find these very small near-infrared emitters that are biocompatible. Ideally, we also want them to fluoresce in this so-called near-infrared 2 tissue transparency window, and that was at the tail end of those graphs, where things like absorption and scattering and autofluorescence were very, very low. We would also like them to be easily targeted to specific biomolecules or tissues, and it would be nice if we could multiplex them. That means have multiple different colors in the near-infrared, so we could look at multiple targets at the same time. And in order to do this, my group is studying an entirely new type of fluorophore called a metal nanocluster. What are these? Well, these are the smallest of metal nanoparticles. You can think of them as little embryonic pieces of metal. We typically talk about their sizes in the terms of atoms, number of atoms, rather than physical size. They contain so few atoms that they're about 2 nanometers or less in size, and they contain about 100 nanometers or 100 atoms or less. And at this size scale regime, due to effects like quantum confinement, stuff gets very weird and exciting. So they can do things that normal materials and even normal nanomaterials cannot do. And one of those things is emit light. So these nanoclusters can be very bright fluorophores, and their sizes and shapes can be tuned extremely precisely. They can actually be synthesized to atomic precision, more like molecules than like materials, where they have defined structures just like a molecule does. And this extreme control over size and shape will give us extreme control over this fluorescence process that we're interested in. Now, a key to controlling the size and shape here is how we engineer what's on the surface of these clusters. So by themselves, nanoclusters are typically unstable. They would rather aggregate, oxidize, or otherwise degrade. But we can protect them with things like molecules on the surface that will shield them from doing that. And those surface molecules are also incredibly important for selecting that size and shape, which, just like for other types of nanomaterials, will strongly tune their properties. So this is the type of process that we're interested in gaining control over so that we can make these nanoclusters both good near-IR emitters and biocompatible. Now, I will mention nanoclusters have many exciting applications well beyond bioimaging in things like catalysis, photonics, potentially for solar energy applications, and for quantum computing. But now let's consider using them as bioimaging agents. So if we can create a nanocluster with a certain size and shape that emits in this near-infrared window, then maybe we can use that as our light-emitting element for a contrast agent. But there's more than just having that atomically precise nanocluster core that we have to consider. There's another level of complexity when we go into a biological setting, and that's related to what we put on the surface. So now what we put on the surface, those surface shielding molecules, doesn't just have to determine the size and shape of the atomically precise core. It also needs to be water soluble, it needs to be biocompatible, and we may need to chemically functionalize it so that we can target that light-emitting element to parts of the body that we're interested in. So that by itself is inconvenient, but there's a whole nother level of difficulty here. And that's that we are now going to put this in the living environment. So we also have to now care about whether that is going to be cytotoxic. We may need it to clear the body in a certain amount of time, for example, through the kidneys. And we also need it to be stable at ionic conditions that match blood, which is very different than the ionic conditions that most synthetic chemists are working with. It's really inconvenient. And then finally, we have to care about how these will interact with biomolecules in the body. And this is a really big challenge, and there are a lot of very talented nanocluster chemists solving this by starting with those systems that I showed you before that have been developed with various small molecule ligands, and then attaching things like this peptide you see here that can target a gastric tumor. But this is very hard, and it often doesn't succeed. So my lab studies a different approach, which is, can we use biomolecules themselves as the template for these nanoclusters and also as the targeting agent so that we don't have to worry about whether this surface chemistry is going to be biocompatible? And there's actually precedent for doing this. It's not a completely crazy idea. Nature has done this for a very long time. So in all of us, there are these iron-sulfur clusters that are scaffolded in complex protein structures. And these protein structures, if you look at the zoom in on the bottom, will present towards this little cluster of iron and sulfur just the right little chemical groups to hold it into place. And these clusters are essential for the metabolic pathways that occur in almost all living organisms. So we would like to do something like this, but with a type of metal nanocluster that will be fluorescent. And so what my lab does is we use DNA and silver. So we use silver because silver nanoclusters are known to be very brightly fluorescent. And we use DNA because DNA is known to have sequence-dependent interactions with silver ions. In other words, little silver ions can go and decorate the nucleobases of DNA. So it was discovered exactly 20 years ago that if you mix silver nitrate, which is a silver salt, and single-stranded DNA in a test tube, and then you chemically reduce it, which is the chemical reaction that you do to form nanoparticles, you can actually form brightly fluorescent nanoclusters instead. And that's because the DNA is wrapping up this tiny cluster of silver rather than letting it grow into a bigger non-fluorescent particle. Now, these nanoclusters are really exciting not only because they're soluble in water and naturally wrapped up in DNA, but also because the DNA molecule itself is controlling the optical properties in a way that is sequence dependent. You can think of these as little tiny nanoclusters with a genome. So the sequence of ACGT that goes into the test tube is going to determine the way that this is going to glow. And you can imagine if we could learn these sequence to property rules, then we could create custom nanoclusters for very specific biological applications. And so that's the story I'm going to tell you today. For the second half of the talk, I'll tell you two short stories. One about how my lab is using high-throughput experiments and machine learning to design near-infrared emitting nanoclusters. And then the second is a mystery that we've solved that actually led us to an exciting discovery about their biocompatibility. So let's get started with what these DNA stabilized nanoclusters are. I'm going to call them AgDNAs for short. So these come in a wide range of different sizes and shapes, and that means that they have a wide range of different optical properties. I've colored some of the dots here in the color that it would actually glow to your eye if you could visibly see it. So you can see they come in a rainbow of colors from blue-green out into the near-infrared, and that size of the cluster is correlated to the glow color, and it's DNA sequence that primarily determines where a nanocluster will sit on that chart. In terms of their properties as fluorophores, they can be very bright. So they have what's called a high quantum yield and a high extinction coefficient in many cases. They emit a lot of light. And they have relatively narrow line widths, which means that we could do this multiplexing relatively easily. Now, two exciting things happened around the time that I started my faculty position here at UC Irvine. And one is related to the discovery of an entirely new set of these AgDNAs in the near-infrared window. So prior to a few years ago, there were very few of these AgDNAs reported with near-infrared wavelength emission. That's above that dashed line here. And it was initially thought that maybe that was just because DNA wasn't good at stabilizing nanoclusters that had the right size and shape for this emission wavelength. But actually, it turned out we weren't looking with the right eyes. So my former lab mate, Stephen Swayze, used a different type of detector, an indium gallium arsenide detector that's specialized for near-infrared emission, and he found all of these near-infrared emitters hiding in plain sight. And this is exactly in that tissue transparency window where we hope to find near-infrared emitters for bioimaging. And then the second really exciting thing that happened was a fundamental breakthrough for these AgDNAs, and that was the determination of some of their atomic structures by several groups around the world. So these were obtained by a technique called X-ray crystallography, and this allows us to localize individual silver atoms and the DNA strands that wrap them up. So this gives us the first pictures of how DNA and silver interact to make this fluorescent nanocluster. So we wanted to take this new information and combine it to potentially design near-infrared emitting nanoclusters, but there's a challenge here, and that's that there are many different DNA sequences we can choose from. If we have a sequence of ACGT, and we have an L number of those, there's 4 to the L ways to combine those. If we have 10 nucleobases, that's over 1 million, but most people in the field were using between 20 and 40 nucleobases. So to put this as my 5-year-old would say, that's a gajillion. And also, there's a really complex connection between sequence and color, and there were really no computational models that could help us solve this problem. So I got interested in this when I was studying these as a PhD student, and I wanted to know, is there another way to do this design? And my question was, could we observe many experimental examples and then learn sequence to property patterns from data? So this is exactly what we also do now in my lab. We have a really nice system to synthesize hundreds of these nanoclusters in one experiment and then rapidly screen their optical properties, connecting DNA sequence to color. We also have the ability for select nanoclusters to really dive into what these are composed of. We can isolate individual atomically precise clusters. We can use mass spectrometry to count up the number of silver atoms and DNA strands. And then we can correlate those to optical properties. And we combine this in order to do what we call chemistry-informed machine learning. Now, what do I mean by machine learning? Well, first of all, I have to give a nod to the fact that last week, machine learning and artificial intelligence had an amazing week in terms of its impact on science. Both the Nobel Prize in Physics and Chemistry involved machine learning and artificial intelligence. These are really powerful tools for science, and these types of models are not explicitly programmed to do a task, but rather learn from many examples of data how to replicate the data, for example. So what we do in my lab is we do many experiments to assign DNA sequence to color, and then we train machine learning models to learn that sequence to color mapping. And we do this in a particular way that is important for how this works. First, we define the problem that we're asking the machine learning model based on the known physics and chemistry of the materials. Second, the way we give the machine learning model that information is also guided by those physical insights, and I'll tell you about that in a minute. And then finally, those models guide our design for new near-infrared emitting nanoclusters, and we can learn what the model learned to push forward fundamental science. So this is a project that has been led by my PhD student, Peter Mastracco, who just got his PhD a few months ago. So he was focused on designing near-infrared emitting nanoclusters, and that's out in that high wavelength regime above 800 nanometers in that plot up there. You can see there's very little data up there. And so our challenge was to use that very little data and amplify the number of clusters in that region. So what we do is we use what we know about the nanocluster chemistry to divide different types of clusters into categories. And then we ask our machine learning model to sort sequences into these 5 categories here. So we can put together a histogram here of the actual number of data points that were in these 5 categories. We only had 55 examples of near-infrared emitting nanoclusters to work with, so this was a challenge. Now, one way that we overcame that challenge was the way that we represent the DNA sequence to the machine learning model. There are lots of standard ways to encode biomolecular sequences from bioinformatics. One simple one is called one-hot encoding, where we turn a sequence of letters into a matrix of numbers that tells us where each letter appears in the molecule. It turns out that this does not work very well for the model to accurately learn sequence to property correlations. And that's partially because this representation makes it hard for the model to learn the underlying chemistry of how DNA wraps up the nanocluster. So Peter's idea was inspired by that same crystal structure I showed you before. He noticed that there were certain molecular motifs for how the DNA wrapped around the nanocluster. And he thought maybe if we represented sequence in a way that could allow these models to learn those types of molecular motifs, that we would be able to better train the machine learning model. So he developed what are called nucleobase staple features, or motifs, that tell us about positions of 2 nucleobases relative to one another. And this turned out to be very important for the success of this project. And then there were a few other things I won't discuss because they will be boring, but I'm happy to answer questions about that. Now, before we deploy this model to design something and actually test it in the lab, which costs time and money, we assess it in silico first. So what we do is we reserve 1/10 of the data, and the model does not see that. We train the model on the other 90% of the data, and we use that unseen data to test the accuracy. And we do this many times to get an average idea of accuracy. So you can see the accuracies here in this heat map. They range from 93%, which is very good, up to 60, or down to 63%, which is not great, but that is the case where we had the most data imbalance because we had very few near-infrared examples, and it's still better than random. So now we use the consensus of all of these models to design new DNA sequences for that near-infrared window. And the results were really remarkable. So we tested 3 different design categories to see how well our model worked. In all cases, the machine learning guided design amplified the likelihood of achieving the specific target color. And the success was especially impressive in the near-infrared window, where we were able to get almost 13 times greater success using this machine learning guided design. And you can see in the histograms on the far side how the color distribution changed. The training data is in the background in gray, and you can see that the machine learning design, which is shown in the front in those colored bars, is shifting that color distribution around into different windows that we're targeting. But not only is this a useful tool practically for design, we can also learn something about how DNA is controlling size and shape. And that's through this process of interpretable machine learning. So we use a process called feature analysis, where we can actually score how each of those staple motifs controls the decisions made by the model. And we found these patterns here, which could be used to design these nanoclusters from a fundamental standpoint, and also give us insights into the chemistry of the clusters. Now, more recently, we worked with my collaborator, who's a computer scientist in New York, to develop a deep learning model to do design of nanoclusters where we have more than one objective. So these are more complicated models, but essentially what they do is they map DNA sequence into a high-dimensional space where those space dimensions are correlated to certain properties that we want to learn, and then we can sample from that high-dimensional space for new design. And this model worked for both shifting the color distribution around and also for making nanoclusters that were much brighter. So this is the type of multi-objective design that we can do with these kinds of models. And these tools are the first models for any kind of predictive design of near-infrared emitting AgDNAs. Now this brings me to the second story. And this story started with a mystery. This mystery was the project of my PhD student, Ana Gonzalez-Rosell. So she was, again, focused on this same crystal structure. And what we wanted to ask was, could we use mass spectrometry to actually count up all the number of silver atoms, DNA strands, and determine the cluster charge of this particular emitter? So we knew from the crystal structure solved by groups in Denmark and Japan that the cluster had 16 silver atoms, shown in gray. It was wrapped up in 2 DNA strands, which are in red and blue. And then there were these 2 silver atoms of what's called low occupancy, shown in pink, which means that sometimes they were there and sometimes they were not in this crystal. So what we expected when we did the mass spectrometry was that we would find a mixture of 16, 17, and 18 atom silver clusters. And I gave Ana this task in February of 2020, so you can imagine that there were some significant delays, but she persevered, and when she was finally able to get into the lab, she brought back this amazingly clean mass spectrum. And each peak here corresponds to a different product, a different number of silver atoms, DNA strands, and at a different charge. So we thought it would be very easy to determine what the cluster was because those dominant peaks are so big. But when we zoomed in, we were baffled. So there was no combination of DNA strands and silver atoms that could give us the mass distribution we observed. And Ana kept repeating the measurement over and over and getting the same result. So what is going on here? Well, we have another element, another type of atom inside of the cluster. But the problem is the periodic table is very big. So there's a lot of possible atoms here. And it took us a long time to figure this out, but the clue actually came from looking again at the crystal structure. And so we asked, are those low occupancy silvers actually silver atoms at all, or are they some other element? And perhaps that other element has 0.3 of the electron density of silver because those low occupancy silvers were there about 30% of the time. And that ended up being the clue that brought us to the answer, which was those 2 little atoms were never silvers at all. They were actually chlorines. And those chlorines attach to the cluster at sites where the DNA strand is sort of separated away from the cluster. Now, we didn't add chlorine at all because chlorine reacts with silver to form silver chloride, and this is not something we want to make. But when we added more source of chloride, we formed more nanocluster. Now, we were convinced in our mind it was chloride, but this was a very unconventional discovery, and so Ana set out on a path of many months to show by many different methods that this was in fact chlorine. So I'll just share 2 of those with you. First, she said, okay, if this is chlorine, then we should be able to go straight down the periodic table and exchange chlorine for bromine because they both have the same number of valence electrons. And so in fact, she could show that that was true. She could show that she could exchange chlorine for bromine, and she would also shift the optical properties of the nanocluster. Next, what she did is she went back to this crystal structure, which had then been studied multiple times by this other group, and they had shown they could mutate individual nucleobases and form essentially the same cluster. And so we thought if the cluster is the same, that means those 2 must also have those mystery chlorides. And in fact, that was the case. There was a lot of work that went into this one slide, but she was able to synthesize all of those mutants to atomic precision and identify chlorides in those as well. So Ana's discovery did 2 important things. One, it advanced the fundamental science of these AgDNAs. So her findings enabled the first ever all-atom computational models to study what's called the electronic structure of these nanoclusters. That provides us insights into what is actually the process going on when they absorb light and then emit light. And these kinds of models had never been possible before. And the second thing, fundamentally, that was done is it gave us a real, true atomic picture of the structure of this nanocluster. We were able to collaborate with the crystallographers who reanalyzed the structure, and they found a perfect match, and you can see some of that structure there. But there was also an unintended discovery here. And that was related to the fact that these chlorides were found to make the nanoclusters much more biocompatible. So we found over the course of studying this cluster for almost 2 years that this chloride-stabilized AgDNA was much more stable in a variety of different solutions than many of the ones we studied. And we started to wonder if maybe the presence of the chlorides was important for that. And in fact, the stability of this cluster was really significant. It actually enabled us to collaborate with Professor Marcus Selden here in the School of Medicine for in vivo imaging in mouse model for the first time. So you can see here in these mice that are happily sleeping that the nanoclusters have been injected into 2 different sites, and what you're seeing is the near-infrared light coming through the mouse tissue and the fur, which was actually brown in this case. So these chloride ligands were what enabled this cluster to be stable enough for these type of experiments, even over several hours. And moreover, we started screening in high throughput for this kind of stability, and we've found since more of these chloride-stabilized clusters. So this now provides us a roadmap for not only how to engineer the DNA sequence that wraps up the nanocluster, but also how to engineer the surface chemistry for stability in biological applications. And this leads me now to where we're at today. So we've made now fundamental discoveries about the nanocluster chemistry, and we've developed a design tool. And this has now set us off running to try to design these as practical tools for bioimaging. And we're really grateful for the support of several different agencies to do this. Now, this is not just work that's happening in my lab at UC Irvine. There's also scientists around the world working on this. And several different breakthroughs have happened, including at the University of Copenhagen, where they developed ways to tag these nanoclusters with different biomolecules and selectively stain individual parts of a cell. And this could be used for this type of targeted tissue labeling of different tissues, organs, and tumors, for example. They also discovered a way to incorporate these nanoclusters into what are called liposomes. These are little lipid nanoparticles, and use this to do blood flow tomography in the brain of a mouse. So these are 2 demonstrations of these practical bioimaging applications with these nanoclusters. We're also collaborating with many experts in photochemistry and photophysics. So for example, with my collaborator, Professor Olesiak Banska in Poland, we've been studying a process called 2-photon fluorescence, where you use 2 long wavelength photons, typically near-infrared photons, that can penetrate deep into a tissue to excite fluorophore. And these nanoclusters have shown remarkably high 2-photon cross-sections, which is even more promising for their use in deep tissue imaging. Similarly, they also have shown promise for a process called upconversion fluorescence, where similarly you can use long wavelength light to excite fluorescence that happens at a shorter wavelength. And all of this could enable us to image deep inside of a tissue. And then finally, this brings us back to our vision. So we are very excited about the potential of these AgDNAs to solve all of these challenges here that have eluded the field of near-infrared bioimaging for some time. So what's going on in my lab right now, we're working on a variety of things to make this vision possible. One thing we're working on is tagging these nanoclusters with different biomolecules that can then target specific molecules, cells, or tissues for practical deep tissue imaging. We're also studying with a collaborator at Northwestern how these nanoclusters behave in living animals. So where do they go in the body of the animal? Do they have problematic toxicity? What types of tissues do they label or not label? And all of this will be important for their practical translation to biomedicine. We're working on how to put those chlorides there on purpose. So those 2 mystery atoms that caused us such a headache turned out to be very important, and we're working now to do that intentionally. And then finally, we're continuing to advance these machine learning design tools and to work with theoretical chemists and physicists to better understand these materials so we can drive forward their development with both experiment and theory. So my long-term vision for this research is that we're going to use advances in fundamental nanochemistry in order to create a new bioimaging technology that could use non-hazardous near-infrared light to image deep inside of biological tissues. I will say this is not a direction I expected. I started out as a physicist, really just interested in stuff that glowed. And it was just this, curiosity-driven research and also my interest in hopefully using my work to make people's lives better that brought us to this place. And the different applications that we envision, there are many. So deep tissue biomedical imaging could impact many different fields of biomedicine, from the study and treatment of cancers to cardiovascular disease to neurology. You might remember at the beginning of the talk, I showed the light scattering in the brain. So brain tissue is very challenging to image in. This is one of these holy grails of imaging that perhaps could be solved with near-infrared deep tissue imaging. We also envision that these types of tools could further fundamental biological sciences, things like neuroscience or systems biology, where you want to understand how different tissues interact with one another. And perhaps if you could tag what's enabling that interaction and watch as it's happening in real time, we could gain more understanding of these types of processes. And then finally, a third area that I'm excited about is the possibility to help people who are developing new therapeutics. So if you have something which is small, bright, and easy to target, what if you could label a new candidate drug and actually watch in real time as this drug is moving throughout the body, rather than relying on the really time-consuming and challenging methods that these bioscientists and engineers rely on today. So these are some of the things we envision. I, of course, don't know exactly what is going to happen, but what I do know is that I don't know where it's going to end up, because I started just being fascinated by light and how it interacts with matter and living things. And so this kind of curiosity-driven research is something that I think is truly important for solving big challenges in our world, including in the biological sciences. So that brings me to the most important part, which is thanking all of the wonderful students and postdocs I work with. You can see some of their faces here. I talked specifically about work done by Peter and Anna in my group, but we also have a list of wonderful collaborators. We work in a very interdisciplinary space using tools from physics, chemistry, computer science, and biology. And this all requires also a lot of collaboration, which is really fun. And then I also am indebted to our funding agencies who make all of this research possible. And I'm finally really grateful for all of you who've taken time to learn about this, and I really look forward to answering any questions. Thanks.